Hello everyone! Last class we went through all of glycolysis. As a reminder, the final product of the splitting of glucose is two pyruvate molecules. As you can see here on the right figure, pyruvate has 3 carbons. Remember, glucose has 6, so during the process of glycolysis, 2 molecules of pyruvate are produced from 1 glucose molecule. Remember, pyruvate is found in the body as the anion. We can see it here with carbon double bound to an oxygen and single bound to an oxygen that has a negative charge. Now you should be able to recognize by looking at pyruvate that this molecule has a terminal functional group. For example, on the one end of the carbon double bound to an oxygen and a single bound to an oxygen anion, we have a carboxylic acid group with the hydrogen dissociated, leaving a negative charge, or a carboxylic acid. My question for you is, what other functional group do you see? The answer to that should be a hydroxyl group, because on pyruvate we also have a hydroxyl group at the second carbon. This figure showcases the 4 pathways, or fates, of pyruvate. The fate or pathway it takes is dependent on the energy status of the cell and what is needed by the body at any given time. Pyruvate at the top of this figure can be converted to lactate if there is a high rate of glycolysis, to acetyl-CoA and onward to the citric acid cycle if oxygen is present, to oxaloacetate if there is a low amount of oxaloacetate in the citric acid cycle, or pyruvate can be converted to alanine if the levels of this non-essential amino acid are low in the body. You need to be aware that although we talk about the glucose oxidation, we think that all the processes involve glycolysis, the citric acid cycle, and the electron transport chain. However, it doesn't always happen this way. When pyruvate is made as a product of glycolysis, that pyruvate will sometimes continue on into the citric acid cycle, which we've already talked about requires oxygen. But we've also seen that it can be converted to lactate if oxygen is not available. Pyruvate can also have 2 additional fates. First, it can be converted to alanine if alanine levels in the body are low. So the question is, well, what is alanine? Well, as you know, and you've memorized from your protein module, alanine is an amino acid. Is alanine an essential or non-essential amino acid? And even if you don't have the essential amino acid memorized, you can deduce the answer to this question by what's happening here. The answer to this is if we are making alanine in the body from a non-alanine precursor, such as pyruvate, that would make alanine non-essential. Your body is actually producing it. The 4th possible fate of pyruvate is that it is converted into oxaloacetate, and you will soon see that oxaloacetate is an important intermediate in the citric acid cycle, and if it runs low, the cycle itself can slow down. So levels are kept high by this reaction, and so we convert pyruvate into oxaloacetate to ensure that the levels are kept at a right amount so the citric acid cycle happens at a fast enough rate. The 2 main factors that you need to be aware of that determines the fate or direction that pyruvate will go is, first off, the requirements of the cell. What is needed by the cell? Second of all is the demand for ATP. What is the energy status of the cell? How much NAD we have, as well as how much our demand for glucose. Do we need to produce new glucose? We are now going to look at the 2 main fates of pyruvate. Firstly, pyruvate to lactate, also known as alactic glycolysis or anaerobic glycolysis. As we can see in the figure, pyruvate gets easily shuttled to lactate. It happens in the cytoplasm. It's a 1-step reaction. The second main fate of pyruvate is pyruvate's conversion to acetyl-CoA through the pyruvate dehydrogenase reaction. And here, as you can see, outlined in yellow, or yellowish orange, is pyruvate needs to move into the mitochondria. Once pyruvate is in the mitochondria, it then gets converted to acetyl-CoA. So there's a couple steps involved. Pyruvate always goes to acetyl-CoA but it's limited by the activity of the enzyme pyruvate dehydrogenase. Now let's talk about lactate formation. This figure highlights lactic acid fermentation. So we start with glucose. It goes through the process of glycolysis to yield 2-pyruvate. During glycolysis, we need to input 2-NAD, which yields 2-NADH. Once we have our 2-pyruvate, we can convert it to 2-lactate through a reaction in which we require 2-NADH, which then produces the 2-NAD. And because this is in the cytoplasm, the 2-NAD go back up to glycolysis and provide the substrate, the coenzyme needed for glycolysis. So NAD is an essential substrate for glycolysis. Without it, step 6 of glycolysis, which needs NAD, cannot happen. So glycolysis is as fast as the amount of substrate it has. As the demand for ATP increases with higher intensity exercise, there is more lactate produced in order to regenerate the NAD for glycolysis to run, instead of waiting for NAD to be transferred from the mitochondria and the electron transport chain. So at high intensity exercise, there is a greater need for NAD for glycolysis to run, and so more lactate is produced to create the cytoplasmic NAD required for glycolysis. It is coincidental that lactate increases. So increased exercise intensity is proportional with an increase in lactic acid formation due to the need to recycle and produce NAD for glycolysis to run. This is called coincidental acidosis rather than causal. Instead, most of the hydrogen ions that are produced are coming from ATP hydrolysis when we break down ATP. Remember, right now, write down that reaction. Write down the ATP hydrolysis reaction, and you'll see that hydrogen ions are produced. That is what contributes to acidosis and not lactic acid formation. Remember that lactate at rest and exercise is going to be different based on what you just learned. At rest, there's not a huge flux through glycolysis, and so we don't need to produce very much lactate in order to produce NAD for glycolysis to run. But as we increase the demand for energy supply to the cell, we need to increase the rate and flux through glycolysis, and as such, we're going to have more lactate formation, and therefore, we can create the NAD required for the glycolysis to run. But at the same time, we're accumulating more lactic acid. So why is this called anaerobic glycolysis? Well, it really is a misleading title. It has nothing to do with oxygen content of the cell. This is all independent of oxygen because there's always lots of oxygen in the cell when glycolysis is running. So although glycolysis does not use oxygen directly, the rest of glucose oxidation does, as does the regeneration of NAD. So both glycolysis and the citric acid cycle run independent of oxygen input. But the citric acid cycle runs by using NAD that's created from the electron transport chain. Remember, the electron transport chain requires oxygen to run. So if oxygen is unavailable, glucose oxidation, the whole process, will get stopped at glycolysis. The citric acid cycle will be slowed down because there's not enough NAD from the electron transport chain. The NAD needed for glycolysis to run will end up getting depleted as glycolysis takes place. And as such, the production of lactate ensures regeneration of oxidized NAD from reduced NADH, which is generated during glycolysis. So the whole process is to ensure that glycolysis can keep running and will generate at least some ATP to sustain rest or exercise. To showcase this relationship a little bit more, let's look at the figure. On the y-axis, we have intracellular pressure of oxygen in millimeters of mercury. And on the x-axis, we have work rate, exercise work rate, as a percent of your VO2 max. So when you start from 0 to 20% of your VO2 max, we see a sharp decrease in the intracellular pressure of oxygen. But from 20% of your VO2 max to 100%, there's hardly any change in intracellular pressure of oxygen content. That means you're never really solely anaerobic. Your cell always has enough oxygen. So it's a very misleading assertion is that the lactate threshold represents the point where the muscle ATP yield from oxidative phosphorylation is compromised by insufficient oxygen, and that maintaining work output requires the addition of anaerobic glycolysis as a source of ATP. Thus, the lactate threshold has been defined as an anaerobic threshold. However, this reasoning does not agree with what we know. Your cell is never fully anaerobic. Instead, it has to do with lactate accumulation and the rate at which we need to produce ATP through glycolysis and the need for NAD for glycolysis to meet the ATP demand. So ultimately, it has nothing to do with oxygen available, rather the rate or speed through glycolysis and the need for NAD for glycolysis to meet the ATP demand causes lactate to accumulate. Lastly, I want you to see that the lactate dehydrogenase enzyme that converts pyruvate to lactate has a lower Km than pyruvate dehydrogenase Km. That means the conversion to lactate will happen at a faster rate and with less pyruvate substrate needed. So it has a lower Km than pyruvate dehydrogenase in the conversion to acetyl-CoA. Now let's look at the biochemical reaction that converts pyruvate to lactate. You can see here that both pyruvate and lactate are 3 carbon molecules. The difference between them is that lactate has 2 more hydrogen atoms. The donor of these hydrogen atoms is NADH, plus a hydrogen proton. So the reactants, or the substrates for this reaction, are pyruvate and NADH plus a hydrogen proton. The product is lactate and NAD. So pyruvate is becoming hydrogenated. We are adding hydrogens to it. So the biochemical process would be hydrogenation, also known as reduction, and the enzyme required would be hydrogenase or reductase. So therefore, the possible enzyme name based on our naming principles that we learned in class would be pyruvate hydrogenase. The actual enzyme name for this reaction is lactate dehydrogenase, since enzymes are always named for the reaction going in the direction of dehydrogenation rather than hydrogenation. This is just one of those naming rules that you'll have to remember, that ultimately that enzyme is always named for the reaction going in the direction of dehydrogenation rather than hydrogenation. So it would be named in the opposite direction, and therefore the enzyme name is lactate dehydrogenase. The bottom line is that lactate is not bad. Lactate production is necessary and a benefit in 2 major ways. First of all, it ensures regeneration of the required substrate NAD, that coenzyme, for glycolysis to run and continue running. Second of all, lactate can be converted back to glucose as a substrate for glycolysis through a process known as gluconeogenesis and the Cori cycle, which we'll talk about on the forthcoming slides. There's a few things I want to touch on again. Remember that lactate formation has little change to the pH or acidity of the cell. ATP hydrolysis is the major contributor to acidity within the cell during exercise. Remember that LDH, lactate dehydrogenase, the Km is less than that of the pyruvate dehydrogenase Km activity, and therefore more lactate will be formed at higher intensity because less substrate of pyruvate is necessary to activate this enzyme. Lactic acid dissociation is not the primary cause of fatigue during max exercise. Acidosis has only been attributes to approximately 10% of the reduction in force production. There's many other possible causes of fatigue, which you've learned about in exercise physiology. There is mounting evidence to show that hypoxia or low muscle oxygen is not the primary cause of lactate production, and that lactate could in fact be used as a source of fuel by numerous tissues, including both resting and contracting skeletal muscle, which we'll talk about on the next slide. Here are some of the major organs that use blood lactate and convert it through the process of gluconeogenesis, the process of creating new glucose, and then put it back into the blood to be used by working muscles and other organs. This is known as the Cori cycle. So the combination of gluconeogenesis, using lactate and converting it to new glucose, and then the process of dumping it into the blood and circulating it around to other tissues to use again is called the Cori cycle. So as you can see here, we have the liver, the heart, the kidneys, as well as other muscles can utilize blood lactate. So we see the skeletal muscle producing lactate. It then is converted into blood lactate. It empties through channels into the blood as blood lactate. That blood lactate can be taken up by the liver and converted to pyruvate. Pyruvate can then go to glycogen in the liver to be stored for another time, or it gets converted to glucose, which then goes back into the blood. We also have the heart, where we have lactate can be used as a fuel source through the process of gluconeogenesis, converted back to pyruvate, and then used in the TCA cycle to yield more ATP. We also have the kidneys, where lactate gets converted to pyruvate, pyruvate to glucose, and then the glucose is used by the kidneys. Any excess is put back into the blood through the Cori cycle, is utilized by any other organ in the body and cell. And then we have other muscle, which it converts the lactate back to pyruvate, and then it can go into the TCA cycle or the citric acid cycle and used to produce energy. So with endurance training specifically, we can increase the number of cell membrane lactate transporters, which allows us to clear blood lactate faster and use it as a fuel source. And so this is one reason why the lactate curve shifts to the right with endurance-based training. This slide describes the pathways of lactate metabolism. That lactate moves from the cell into the blood through a transporter, and this is known as MCT transporters, and they have 1 to 4 isoforms. MCT stands for monocarboxylase transporters or transferase. We typically see MCT1 for lactate, and lactate can be converted back to glucose through the process of gluconeogenesis, the creation of new glucose. So in the liver, we have gluconeogenesis happening, and then we have the Cori cycle, which is essentially glucose traveling in the blood to other cells for which it can be used as an energy source. In the liver, we can also store an excess amount of glucose in the form of glycogen. The heart, being a highly oxidative tissue with many mitochondria, uses lactate as an energy source. It converts it back to pyruvate for use in the citric acid cycle. In the kidneys, we have gluconeogenesis happening, so we produce pyruvate back to glucose. That glucose then gets dumped into the blood, and via the Cori cycle, it gets delivered around the body. And then we have muscles, so slow twitch or type 1 muscle fibers are well adapted to utilize lactate as an energy source. So we just explored one of the 4 pathways for which pyruvate can travel, specifically the formation of lactate from pyruvate, which is done in one simple biochemical reaction that requires NADH and creates NAD with the enzyme lactate dehydrogenase. The NAD is needed for glycolysis to run and yield ATP. So in the next video, we will explore the conversion of pyruvate to acetyl-CoA and more about the citric acid cycle.